Hello everyone, my name is Karen.
And my name is Hanisa.
We are excited to present to you our research on hydrolase gene expression and analysis.
Our goal was to express, extract, and analyze the gene encoding for a hydrolase enzyme.
Before we dive into our research, what are hydrolases? Well, hydrolases are simply enzymes that break down molecules using water. They're responsible for breaking down carbohydrates, proteins, and lipids, among other molecules.
Methods. Our research began by reviving frozen DH5 alpha cells that carry the gene of interest in a plasmid with the encoded hydrolase enzyme. We then isolated the plasmid from DH5 alpha cells since this cell type is not made for large-scale expression investigations. We placed the isolated plasmids into BL21 DE3 E. coli cells to maximize protein expression. We grew a large quantity of BL21 DE3 cells containing the gene of interest and conducted autoinduction to further maximize the expression of the gene. We extracted the protein by breaking BL21 DE3 cells open and calculated the total amount of protein employing the BSA standard curve.
We then purified the protein using metal affinity chromatography in the form of fractions. We identified the fractions that have the expressed protein to examine its purity through the SDS page in Comasi blue staining. Finally, we conducted a hydrolase assay to test whether the protein is an enzyme, and enzymatic activity based on the temperature was measured.
Results and discussion. Let's now discuss the results. As you can see, we aimed for a more visual representation of our data for our audience. The grown DH5 alpha cells on LB agar plates with ampicillin. These cells are specialized for storing plasmids but not expressing gene of interest. Ampicillin was added to the culture for the best survival of culturing cells during our experiment.
The Bradford protein assay. This was completed to measure the presence and total protein concentration from the BL21DE3 cells that we grew that contained our gene of interest.
The results of the Bradford assay showed that we had successfully extracted and grown a substantial amount of protein from the BL21D3 cells. The concentration of protein was measured to be around 15.96 micrograms, which was sufficient for further analysis.
The hydrolase activity assay. The SDS page and Comasi blue staining showed consistent results with the hydrolase activity. However, it was interesting how fraction 1 and 4 had the most activity compared to the rest of the fractions. We concluded this was mostly due to the way we pipetted the fractions to the cuvettes.
We found that fractions 1 and 4 show the highest levels of hydrolase activity, which was confirmed by both the Comasi blue staining and the activity assay. These fractions were further analyzed to determine the purity of the expressed protein. The Comasi blue staining also confirmed that the protein in these fractions was highly pure with no visual contamination from other proteins or impurities.
The hydrolase temperature assay. As temperature increases, so does the rate of enzyme reactions. Most enzymes will increase activity by 50% to 100% after 10 degrees Celsius rise. However, the increase is only up to a certain point until the elevated temperature breaks the structure of the enzyme and becomes denatured. Based on our results, our enzyme's optimal temperature was around 60 degrees Celsius, which would suggest it has great thermostability. Speaking of thermostability, it makes me want to wish I was a hydrolase. In the summer, I decreased by 200%.
The temperature assay showed that our hydrolase enzyme was highly thermostable, with optimal activity at around 60 degrees Celsius. This is an important finding as it suggests that this enzyme could be used in a range of high-temperature applications. In conclusion, our research highlights the importance of studying hydrolases and their potential uses in medicine and, yes, in industries and environment. While our results were promising, there are still areas for improvement in our experiment. With that said, thank you for tuning in with us.
Until next time.